Hello, everyone. Let's get started. My name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. I'd like to make a few points about today's seminar. In support of the next generation of cancer researchers, today's Cancer Moonshot Seminar will feature research from talented early career scientists. These are abstract-driven talks nominated by Cancer Moonshot investigators and selected by NCI staff. Before we start, a few bookkeeping items to review. Questions will be responded to at the end of the second presentation. Please enter your questions in the Zoom Q&A box. Please do not enter in the chat box. The audience can prioritize the questions by hitting the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions are also provided in the chat box. The recording of today's webinar will be available a few weeks after today's presentation. With that, I'd like to introduce our moderator, Dr. Anand Patel. Dr. Patel is an assistant member and pediatric oncologist at St. Jude Children's Research Hospital. His research interests are focused on pediatric solid tumors, where he uses 3D cell culture models and single-cell sequencing technologies to understand mechanisms of treatment resistance and recurrence. He is one of the co-leads for the Human Tumor Atlas pilot project, which is setting out to comprehensively map heterogeneity in neuroblastoma using both single-cell sequencing and spatial omics tools. Dr. Patel.
Thank you very much. I'm looking forward to today's talks. We have two great speakers today, so I'm going to go ahead and introduce our first speaker, Dr. Jeremiah Walla, who is a computational biologist and a clinical fellow in medical oncology at the Dana-Farber Cancer Institute and Mass General Brigham Cancer Center in Boston. His graduate work focused on sequence assembly methods for identifying structural variants and their significance in cancer whole genomes. His current work uses spatial profiling methods to answer translational questions in both GI and GU cancers. His clinical focus is on treating patients with genitourinary cancers. So Dr. Walla, we're looking forward to your talk. Take it away.
Great. Thank you, Dr. Patel. It's an honor to be here and looking forward to the discussion. So we'll talk about our work on spatial profiling in colorectal tumors and trying to build some translational results here. So just an overview in one slide of how we treat colon cancer to just frame the discussion for the types of problems that we're trying to solve. So for tumors that are resectable, stage 1 through 3, the standard of care is upfront surgical resection. And then depending on the TNM stage and then the number of risk factors, which I'll describe in a moment, patients can either be sent to just observation and then all the way up to 6 months of doublet chemotherapy. For advanced or metastatic disease, for the majority of patients that are mismatch repair proficient, standard of care is doublet or triplet chemotherapy with or without a targeted agent. And for the minority of patients that are mismatch repair deficient, the new standard of care is now first-line checkpoint inhibitor immunotherapy. Getting the balance of all these therapies is incredibly important, not only because, of course, we want to make sure that our localized patients have the best shot at cure as possible, but also we want to avoid over-treatment, and specifically oxaliplatin neuropathy is a significant burden and a lot of a driver of a reason why we need better biomarkers in colon cancer. So what constitutes high-risk localized disease? If you look at the NCCN treatment guidelines, they stratify a number of different features that clinicians consider. Among the most important are histopathological features like poor differentiation or markers of a highly invasive tumor. Clinical characteristics such as undersampling of lymph nodes or bowel obstruction are important. Interestingly, in colon cancer, there right now is insufficient data to support the use of multi-gene assay panels, despite there having been around for many, many years. And this is quite different than several other tumor types, including breast cancer. Circulating tumor DNA is exciting. It's emerging. Stay tuned, only used right now in a clinical trial setting. So among imaging-based methods, Immunoscore is perhaps the most well-studied. And this is from our European colleagues. And essentially, it's a digital histopathology on CD3 and CD8 stains from TMA tumor microarrays from tumors. And you can use these stains to then create a score where you can stratify patients as high, medium, or low. And then a really nice cohort study here, they found that patients with high immunoscore profiles tended to have better disease-free survival in the localized settings. If you look at the European guidelines, they suggest that immunoscore could be helpful for refining prognosis, but its role as far as predicting benefit for chemotherapy is uncertain, and they specifically point to a need for further studies, specifically within the stage 2 cohort, where this is perhaps among the more important questions to be answered. So why spatial profiling as a biomarker in colon cancer? Well, first is morphology matters. We've known for over 100 years that histopathological review of how tumors look under a microscope has been the gold standard for diagnosis. Tumors are certainly heterogeneous. And then tumors are both their cancer cells and the surrounding stroma. And really over the past decade, we've had more and more success specifically targeting stromal component of tumors rather than necessarily the cancer cells themselves. So having an assay that captures both compartments here, I think, will be incredibly important as we continue to develop those therapies. So how we do it, immunohistochemistry is a form of spatial profiling. It's cheap, it's fast, it's part of the clinical standard practice in many cases, but it's very targeted. You have to know exactly what you're looking for. Spatial transcriptomics, I would say, is the flip of this. It can be very high plex, up to a whole genome, but more expensive, best used on frozen tissue, not quite single cell yet, and really important in a research context. I would argue that multiplex immunofluorescence, which is what I'll talk about, straddles a medium space between these two, both in cost, but in its plex as well. But it's also single cell, and I think incredibly importantly, it's used on FFPE-based tissue, so tissue that's part of normal clinical workflow or even archival specimens can be profiled. In particular, the technology we use is something called Orion. It's a variant of our SciSIF platform. And Orion was really developed by Jerry and colleagues in our lab to address more translational questions. So the idea is that you want to minimize the amount of physical labor required to do this immunofluorescence staining. So this is all done with a single pass. We can pack 16, 20 different fluorophores on this one pass. And then importantly, once you're done with it all, you can do a clinical grade H&E staining on that exact same specimen. So the idea of putting this into a clinical workflow becomes a lot more palatable when you have the gold standard diagnosis that can still come out at the end of all of this. What these images look like is I think, quite beautiful. In the background of this is the Orion multiplex image, and then overlaid on top of this is the same section H&E image, and using one of our viewer tools, you can sort of slide back and forth between the two different views. So I'll focus the rest of the talk on one particular project of ours, which is a multimodal imaging atlas of 74 colorectal tumors. These are all whole tumor upfront resections. It's a retrospective cohort. We've done 16-plex Orion imaging on this. But I think importantly, and we've really come to understand this more, that really detailed, careful clinical annotations are incredibly important for determining whether what we're seeing in the imaging space, how that impacts the clinical prognosis. We've had MD reviewed progression-free survival curves, and then we have the histopathology reports from all of these patients. And then finally, we have some molecular data. So this is clinical grade targeted sequencing of known cancer driver genes. From that, we can also calculate tumor mutational burden. And then finally, we have the immunohistochemistry for the mismatch repair status. So what sort of problems could something like this address? Obviously, we're interested in this big question of who should get adjuvant chemotherapy, how long should they be receiving it for? Is there anybody who we could de-escalate in? But I think particular to a multimodal atlas, what we've really been interested in is trying to better understand relative effect sizes among different classes of biomarkers out there, molecular, clinical, imaging based. You can also use the different molecular stratifications to then compare how these different tumor groups look like under a microscope. How does the immune microenvironment of, say, mismatch repair deficient compare with that of proficient? And then ultimately what would be fantastic would be an imaging-based biomarker or some biomarker that could predict response to immunotherapy. And in colon cancer, that really would be particularly for the mismatch repair proficient patients who right now there's no known role for immunotherapy, although I'll comment on that towards the end. So looking at our cohort in particular, I think the first thing we can see is that it's fairly similar cross-section as much larger cohorts in colorectal cancer. Our distribution of mutations is quite similar to sort of known distributions out there. And overall mutations are a modest predictor of outcomes. You know, KRAS mutated tumors tend to do a little worse than those that are wild type. And if you look at the forest plots on the right of our cohort compared with the literature, you see that they sync up pretty well. So going towards the imaging, one of the things that we really spent a lot of time on and really focused on getting correct was figuring out exactly where the tumor is on these slides. And it's sort of a basic question, but it turns out to be a difficult problem. Each of these tumors is incredibly heterogeneous. There's different histologies, so there's mucinous, there's poorly differentiated. And so to try and create a completely computational workflow to segment out these tumors is an incredibly difficult problem. So we've favored a combined approach. So for all 74 of these, we've manually annotated the tumor compartment, and that's with the help of some of our pathologists in the lab. And this is really useful for separating out tumor from normal epithelium, which can have a lot of tissue-based markers that look like tumor, but nevertheless are normal epithelium. And then secondly, on top of that, we can do a computational approach where we have a KNN classifier that can identify individual tumor cells so that we can get the finer details of where the tumor compartment is. And it also allows us to trace out this tumor margin between the stroma and the tumor. So once we have those annotations, it really just comes down to looking at what are the features of different cell phenotypes within the tumor compartment. And one of the first things that we notice is that there's a pretty striking range in terms of how T-cell infiltrated tumors are. And so on the left, shown here is just a tumor that's quite T-cell infiltrated. You get these really nice CD3 positive, here shown in pink, scattered all throughout the tumor compartment. And then on the right, there's really a complete absence of T cells. If you just stratify patient outcomes based on this T cell marker within our cohort, you get a really profound separation of the curves of progression-free survival, whether you look at CD3 or CD8. I will say when I first saw this curve in our data set, it just made me think that, well, maybe we're just looking at the mismatch repair deficients. We know that they have a high mutational burden. We know that there's a lot of T cells in those tumors, and we know that they have good clinical outcomes. Maybe this is just reflecting that cohort. So what I'm showing here is a waterfall plot, so you can see the individual tumors that we have, and colored here by mismatch repair status. You know, indeed, we see that most of the mismatch repair deficient tumors are grouping at the top. That's expected. But surprisingly, there's a pretty good subset of the mismatch repair proficients that nevertheless also have a very high CD8 infiltration. So we really focus our subsequent analyses on a slightly more refined subset of these tumors, where we're categorizing this new class we're calling TIP MMR, or T cell infiltrated mismatch repair proficient tumors that are proficient but nevertheless have a mismatch repair deficient level of T cell infiltration. So with this categorization, we looked at, are there any molecular markers that distinguish TIP MMR tumors? The first one to look at is just mutational burden. Could this just be because of neoantigen load? As far as we can tell, that doesn't seem to be the case. There's really no difference between the T cell included and the T cell depleted mismatch repair proficient tumors, and quite different than the deficients. Similarly, you know, even though the cohort's still relatively small, we don't see any particular enrichment for any known driver genes as being associated with T cell infiltration. In particular, we were interested in whether this could just be because of POL-E or POL-D1 mutations, and that doesn't seem to be the case. None of our tumors had those mutations. So, you know, again, we've known that T cell infiltration is a really strong prognostic marker, but what we really want to do is now, using this new categorization, separate out the mismatch repair deficient and just focus on the mismatch repair proficient cohort. And really striking the separation here of the curves based on T-cell infiltration alone still shows this really striking difference in improved patient outcomes with more T-cell inclusion. So we can use the other covariates and other features of our atlas to start to get at effect size estimates for these different categories. On the left is a permutation analysis. So it's basically looking at, what are the possible theoretical biomarkers left out there that still separate out about a third of the patients. And if you do a random permutation on those labels and compare with what we actually see in terms of T-cell infiltration, the TIP MMR as a discriminant really does surface as one of the best discriminants in terms of hazard ratios. Another way to look at this is with the univariate Cox proportional hazards model. So we can compare the relative explanatory power of different features in our data set, and really see that the top level features, the most explanatory is T-cell infiltration in the tumor cells, followed by other histopathological markers, and then just greatly outperforming the known mutational markers. So we have a number of other markers in our panel, of course, other than just T cells. And so we focus next on looking to see if there's any change in the other immune microenvironment composition between these different categories. And we're particularly interested in looking at markers of immune exhaustion and immunosuppression. And here we're looking at FOXP3 positive Treg cells. And shown here on the left, you get this really nice nuclear stain in pink on CD4 membranous staining here, showing Treg cells. And if you compare the distributions of just how many of these cells you get across the different types, you see that the TIP MMRs are really a distinct subcategory here in terms of the density of Treg cells, suggesting that there's potentially a more interactive push and pull within these tumors. Similarly, we're interested in, of course, checkpoint inhibition, PD-L1 and PD-1, of course, being targets for immunotherapy. And really, we see that TIP MMR are much more similar to mismatch repair deficient, both in terms of PD-L1 and PD-1 staining, and are distinguished from the T-cell depleted just in terms of the amount of these markers. And just shown on the right is just an example of, this is actually from a mismatch repair deficient tumor, but showing a really nice membranous PD-L1 stain, very, very dense in this tumor, and then a lot of infiltrating CD3 positive cells as well. So finally, we want to see where these PD-L1 signal is coming from. In some tumor types, it's known to come mostly from tumor cells, like in the lung. In other tumor types, esophageal, we're interested in the combined positive score, so looking at PD-L1 expression on both tumors and myeloid compartments. And so we see across our different categories, the TIP MMR tumors tended to have significantly higher expression of PD-L1, specifically on the macrophage compartment. And not only on macrophages, but if you look at the image, you can see physically where that is, where this is sort of palisading set of PD-L1 positive macrophages really lining the borders of these tumors, suggesting that this is just really an area, again, of a lot of push and pull in terms of T-cell exhaustion versus cytotoxic active T cells. So lastly, because this is a tumor atlas, we really want to make this accessible to the broader community. We want to integrate this with really one of the best atlas structures out there, which is CBio Portal, which of course is focused on categorizing molecular data. So we've worked with Eno de Bruin down at Memorial to integrate now our imaging-based features. So for instance, you can do an oncoprint here on the left with both clinical features, molecular features like single gene mutations, and then also now you can add in density of different tumor, different cells, for instance, CD3 positive cells, as derived from the imaging. And then importantly, for any of these tumors, you can just click on them and it will take you to a link out of this interactive viewer called the Minerva Story, where you can zoom in and out, you can change the different markers, and you can really get a sense for looking at these tumors and finding out sort of where the cells of interest really are. So just in conclusion, we've used our multimodal atlas to identify a subset of mismatch repair proficient tumors. We've termed them as TIP MMR that have improved outcomes, mismatch repair deficient levels of checkpoint markers, and signals of increased Tregs and PD-L1 positive cells. One of the things that we've thought about going forward and have really focused on with these multimodal atlases is just the incredible importance of having very well annotated clinical cohorts. And in particular, we're really interested in applying this methodology to prospective cohorts. So in clinical trials where the population is very highly controlled, there's a lot of help just in terms of getting the annotation out there. And I think it's really incredibly important as we scale these things up to do so in a way where we can actually more readily translate what we find in the lab into an actual biomarker, because we have the clinical data and the appropriate cohort for doing so. Just the very last point I just wanted to raise here is this hypothesis that we've been thinking of is, are these TIP MMR patients good candidates for first-line immunotherapy? We know that in heavily pretreated mismatch repair proficient, that there aren't any responses to immunotherapy. But we also know that the earlier you use immunotherapy, the better. This is across most cancer types. And in particular, point out this early phase study, the niche study that looked at first-line immunotherapy in both MMRD and MMRP patients, and really found up to a third of patients had a pathological response. A majority of those were a major pathological response. And it certainly, I think, begs the question whether the groups of things that we're seeing and others have seen as well, are these the patients who would benefit from immunotherapy? So just want to thank my mentor, Peter Sorger at Harvard Medical School, and incredible support from his lab, Jerry, Yuan, Kristen, and Andrian in particular, the support from Sandro Sanagata's lab, Sabrina Chan, and Shannon Coy, Eno de Bruin down at Memorial, and then our clinical collaborators, Dr. Giannakis and Dr. Parikh. So thank you, and looking forward to any discussion.
Terrific. Thank you so much for sharing your data. Just to remind everyone, we'll have a panel discussion after the second talk, and feel free to put your questions in the question and answer section. We're going to move on to our second speaker today, who is Dr. Seth Zimmerman, who is coming from Duke University, where he's a postdoctoral fellow in the lab of Dr. Chris Counter. His doctoral work focused on the design and implementation of optogenetic tools to study cell migration and metastasis, and he currently uses high-throughput technologies to identify cancer cell vulnerabilities with a focus on rhabdomyosarcoma and pancreatic cancer. So take it away, Dr. Zimmerman.
Yeah, thank you, Nat, for that introduction. So at the top, I'd like to first thank you all for logging in and listening to my presentation. And then I was also lucky enough to be part of this Cancer Moonshot project, in which we were trying to define cancer vulnerabilities for rhabdomyosarcoma, specifically PAX3 FOXO1 or fusion-positive rhabdomyosarcoma. And this was this whole project was headed by Corinne Lenardic, and so I just want to show my appreciation for her as well as the whole group and their input. And so I'm going to tell you about my contribution to this project. And so to set the stage, rhabdomyosarcoma is a cancer, pediatric cancer that's relatively rare, but it's the most common soft tissue pediatric sarcoma. And so it has an overall survival rate of about 70%. However, there's two histological subtypes. There's an embryonal subtype and an alveolar subtype. And the embryonal subtype has a 5-year survival rate of about 80%, while the alveolar is about less than 30%. And so clearly these are two separate distinct tumors, and there's a lot of work to be done to understand and to try to find better treatments for the alveolar subtype. There's a second way of subtyping these tumors, and that's by genetics. And so there's fusion negative and fusion positive, and there's a relatively high concordance between alveolar and the fusion positive subtype. And what I mean by fusion positive is that most of these tumors are driven by fusions either between the PAX3 transcription factor and FOXO1 transcription factor, or PAX7 and FOXO1. However, the concordance between alveolar and fusion positive is relatively high, but there's some cases where they can't identify a fusion protein in the alveolar subtype. And so in many of these cases, what they're beginning to identify or find is that these tumors contain other fusions as well. So like I mentioned, there's a PAX3 FOXO1, PAX7 FOXO1 fusion, and then the more rare PAX3 FOXO4, NCOA1, NCOA2, MAML3, and INO80D. And so all of these different fusions are found in the same sort of cancer, fusion positive rhabdomyosarcoma. But the C-terminal fusions have these different sorts of functions where the FOXO fusions or FOXOs are transcription factors, and COA1 and 2 are histone acetyltransferases. MAML3 is a transcriptional co-activator, and INO80D is the regulatory subunit of the chromatin remodeling complex. And so the question is, how are these different fusions driving the same sort of cancer? And so our hypothesis was that maybe these oncofusions are producing a common transcriptional program through their protein-protein interactions. And so to test this, we took a model of fusion-positive rhabdomyosarcoma called an RH4. We're going to knock down PAX3 FOXO1 with doxycycline-inducible shRNA and then reconstitute the different fusions within these cells to make sure that they rescue growth, the transcriptional output of PAX3 FOXO1. And then we're going to look at the protein-protein interactions of each of the different fusions within the cell to try to find sort of commonality between these different fusions. In order to do this proteomics, we've taken a proximity labeling approach where we express our fusion protein further fused to a turbo ID domain. When we express this gene in the cells, it'll express the fusion protein fused to the turbo domain, and the fusion will bind to its normal endogenous interactions. But when we add biotin, the point of the turbo domain is to activate this biotin, which will then covalently bind to proteins within proximity of the fusion protein. We can then purify the interactions through streptavidin pull downs and identify which proteins are interacting with the different fusions by mass spec. So what we expect to find is that each of these rare fusions will rescue the growth of these RH4 cells when we knock down PAX3 FOXO1. They'll rescue the transcriptional profile, but they'll also share some sort of protein-protein interactions that are producing this sort of function. And so to start, we just wanted to make sure that we were able to rescue the growth of these cells when we reintroduce these different fusions. And so to begin this, we first wanted to make sure that when we knocked down PAX3 FOXO1, we actually inhibited cell growth. So you can see here in that last column, with the addition of an shRNA targeting PAX3 FOXO1 as well as doxycycline, we get reduced cell growth almost to nothing. So these cells have either died or differentiated. However, we can rescue these growth defects by adding the different fusion proteins. You can see here that most of the fusions rescue the growth of these cells to the same extent as a wild type PAX3 FOXO1. However, there's some subtle differences here in the PAX3 INO80D and PAX7 FOXO1 fusions. But we still consider this a partial rescue as the cells continue to grow over time, unlike just the knockout itself. So once we validated that the fusions can actually rescue the growth, we wanted to see how the different fusions affected the transcriptional program of the cells. And so we performed RNA sequencing after rescue, and then analyzed the RNA-seq by principal component analysis, just to orient you to this graph. The gray dots here on the left are the positive control where we knock down PAX3 FOXO1, compared to the black dots where the PAX3 FOXO1 is still intact. And each of the different rescues are different colored dots here. And I've highlighted the PAX FOXO fusions in blue. And as you can see, as you go from the positive control where we've lost PAX3 FOXO1 to the wild type-like cell, we start to begin to rescue that same sort of transcriptional output with the different PAX FOXO fusions. Interestingly, the other fusions produced almost completely different transcriptional outputs at least by principal component analysis, and the PAX3 NCOA1 or NCOA fusions clustered together over here. So when we look at the protein-protein interactions with these different fusions, we see a similar sort of pattern where the PAX-FOXO fusions sort of cluster together, the NCOA fusions cluster together, and the INO-ADD and MAML3 are different. And comparing these two graphs, I think what this is telling us is that there are different transcriptional outputs, and that's actually driven by the different fusion proteins. And so we were sort of surprised by this fact that there's a sort of different transcriptional output and different protein-protein interactions, but principal component analysis is really good at finding differences. And so we wanted to look using some other technique to find similarities between them. And so we used gene set enrichment analysis to look at the transcriptomic profile of these cells. And using the hallmark gene sets, it's a little bit small print here, but hopefully you can see that the E2F targets and the G2M checkpoint pathways are both over expressed for each of the different fusions, while myogenesis transcripts are depleted. And so what I think this means is that with the E2F and G2M checkpoints being upregulated, cells are cycling more rapidly, while myogenesis is downregulated, showing that these cells are being prevented from differentiating as they're supposed to. There's also gene sets that are known to be affiliated with PAX3, FOXO1, or ARMS specifically. So on the plot on the right here, I'm just showing you a control cell that has a scrambled shRNA compared to a knockdown of PAX3, FOXO1. And you can see these different enrichment scores for gene sets involved in PAX3, FOXO1 transcription. For all of the different fusions, we can see similar sorts of patterns in the transcriptional profiles of these different fusions. And so again, what we think this means is that while the transcriptional profiles of these different fusions are different, when it matters, in the areas that it matters, such as the depletion of myogenesis, as well as upregulation of the cell cycle, we see very similar patterns in the gene expression. And when we look at the protein-protein interactions, we see a sort of similar output. And so in total, using the proximity labeling, we identified 2,073 different protein-protein interactions between all 7 different fusion proteins. However, when we look at the overlap between each of the 7, we find a central complex of 410 protein-protein interactions that were found in each of the fusions. And so what I'm going to call this from now on, this is the common interactome, or the fusion common interactome. And importantly, again, we find protein functional units that have been associated already with PAX3 FOXO1 function, fusion positive rhabdomyosarcoma, such as the SWE/SNF complex, the mediator complex, and hippo signaling pathways. And I want you to keep the hippo signaling pathway in mind later for this talk. One of the most interesting findings in this proteomics was that each of these different baits, so all of the different fusion proteins, actually interacted with the C-terminal, the full-length protein of the C-terminal fusions. So for example, PAX3 FOXO1 interacted with FOXO1, NCOA1, NCOA2, MAML1, not MAML3, and INO-ADD. So in cases like MAML3 and MAML1, MAML1 is a close homologue to MAML3 and is able to actually substitute for MAML3. And we find that MAML3 isn't actually expressed in the rhabdomyosarcoma cells, which is why we aren't seeing it in the protein-protein interactions. So together, what we think all this data means is that each of these different fusions are able to rescue the growth of the cells as well as the transcriptomic profile, and that we found a common protein-protein interaction network of 410 common interactions, and that within those 410 common interactions, we actually found the C-terminal fusions, the full-length proteins of the C-terminal fusion partners. And so this allows us to make a model in which PAX3 FOXO1 is able to localize FOXO1 to the PAX3 binding sites within the genome. FOXO1 is able to then bind all of its friends, including NCOA 1 and 2, MAML, and INO-ADD. And then they are also bringing along these other 410 protein interactions. And so the PAX3 with a FOXO1 C-terminal fusion is somewhat fungible, because if you substitute it for NCOA1, you're still bringing along the same 410 proteins, so the same core complex, to the same sites within the genome, causing the aberrant transcription. So another way of saying this is that it's it's not the function of the C-terminal fusion that matters, but it's the proteins that it interacts with. And so this is an interesting model, I think, in itself, but it has some implications field as well. And so one of these implications is that can this common interactome predict novel RMS fusions? And the idea here is that since each of these different fusions, since we found the C-terminal half of each of these different fusions within the 410 protein-protein interactions, is it possible that there's fusions that haven't been identified yet that are also contained within this 410? And so with the help of some collaborators at the Princess Maxima Center in the Netherlands, we actually showed that this is the case. And so a patient there presented with a tumor, a rhabdomyosarcoma in their shoulder that had all of the immunohistochemical markings of rhabdomyosarcoma. But they couldn't find a PAX3 or PAX7 interaction, a PAX3 FOXO1 or PAX7 FOXO1 fusion protein within this tumor. So in order to help them define what type of cancer this is, they used RNA-seq and found that it more closely clustered with embryonal rhabdomyosarcoma, not alveolar, and so a fusion negative subtype. However, using the same RNA sequencing, they found transcripts that aligned to PAX3 WWTR1 fusion. And so this may be a novel fusion for rhabdomyosarcoma. And if you remember, in our 410 protein interactome, we found the hippo signaling pathway, which includes at its center the WWTR1 protein or gene. And so we think that our 410 common interactome is able to help us identify novel fusions within cancer. And so we were able to form a tumoroid model from this patient's tumor and then knock down the PAX3 WWTR1 fusion and show that the same sort of transcriptional output that we saw for all the other fusions was also produced by the PAX3 WWTR1 fusion. And so we think that our common interactome in combination with RNA sequencing may be able to help identify future novel fusion proteins in patient samples. And so a second implication from our analysis is that PAX3 NCOA2 is found in rhabdomyosarcoma and thought to bring this sort of common interactome to PAX3 locuses. But in mesenchymal chondrosarcoma, for example, there's a HEY1 NCOA2 fusion. And so we thought there might be a possibility that this is essentially the same as PAX3 NCOA1 or NCOA2, except that it's driving this common interactome to HEY1 locuses, which is important for mesenchymal chondrosarcoma instead of rhabdomyosarcoma. And so it might be the case that this common interactome is being used in other cancers. And so we again took our 410 protein common interactome and looked in a data set of fusion proteins known to be found in cancer. And we found that within the 410 common interactions, about a quarter of them are contained in at least 2 other fusions known to be involved in cancer. And so the last implication here is probably the most important, and that's the fact that, like I said previously, these tumors are very dependent on the fusion proteins in order to drive the cancer. So you might think this would be an easy cure. We can just target PAX3 FOXO1 therapeutically, and that would help to reduce the tumor, get rid of the tumor. However, that's been tried and it's been proven very difficult to actually target PAX3 FOXO1. So we thought, let's take a step back and look at the proteins that PAX3 FOXO1 interacts with, or that all of the fusions interact with, in the hopes that those proteins are what's helping PAX3 FOXO1 to drive the cancer. So if we look at the 410 common interactions, in order to find a sort of functionally relevant interaction, we cross-reference those 410 proteins with the DepMap database, which is a data set of whole genome CRISPR-Cas9 screens across hundreds of different cell lines. And so we compared all of the different fusion-positive rhabdomyosarcoma cell lines to the rest of the cell lines in the cancer. And long story short, the graph that I'm showing here, the dots on the left, farther to the left, are genes that, when depleted in a fusion-positive rhabdomyosarcoma cancer, inhibit the cell growth. So what's nice to see here is that PAX3 and FOXO1 are both there because we're likely hitting the fusion gene. We also see MyoD1, which is a known interaction with PAX3 FOXO1, and a transcription factor. And so trying to target it would be similar to trying to target PAX3-FOXO1. And so we also find similar specific interactions for rhabdomyosarcoma that deplete cell growth. And so the further research here is going to be to try to actually target these, and we're having some luck in identifying proteins in this from this graph that are actually therapeutically targetable and tractable. And so in conclusion, again, we identified this common interactome of 410 proteins, and with that, we were able to model how these protein fusions function in rhabdomyosarcoma, and that they have the C-terminal fusions are relatively fungible because they're able to still localize the same core set of proteins to PAX3 binding sites on the genome. And second, we think that this 410 might be a nice data set in order to help identify novel fusions in tumors. Third, I showed you that in other cancers that might be taking advantage of this core set of proteins. And fourth, we can use this core set of proteins to try to identify novel therapeutics against fusion-positive rhabdomyosarcoma. And with that, I'd like to thank you all for listening and thank the Counter Lab, specifically Dr. Counter, Lily, who's a tech in the lab, who's done some of the experiments that I showed here today. And then, of course, the U54 group for all of their feedback throughout their process, and our collaborators at the Princess Maxima for their contributions. Thank you for listening.
Thank you, everyone. Feel free to put your questions in the question and answer area. We have plenty of time. I think there are, so far we've had a couple of questions for Dr. Walla. So maybe we'll start with you first. Is that okay, Dr. Walla?
Sure, yeah.
Great. So it looks like we have two questions, and actually I was thinking along the same lines as well. So Andrea Burnett Hartman asked you, as you mentioned, T-cell tumor infiltration is a well-known prognostic biomarker. Is infiltration being used much in the clinic as a predictive model to guide treatment decisions? And do you think that your Orion method might inform ways to make a better, immunoscore down the road?
Yeah, great question. So the first answer is no. It's not being used clinically. If you look at what is used clinically, mismatch repair status, that came out of a trial, the Quasar trial, which is a big, large prospective randomized trial of adjuvant chemotherapy, and they had profiled patients for mismatch repair. So you could go back and you could do the subset analysis, and you could see in a randomized sort of prospective way whether or not this was predictive. So that same thing doesn't exist for something like T-cell infiltration. I think, to answer the question about how Orion or something like this could fit in, I think what we really need to be doing is really focusing on analysis of prospective trials, of things where we can see the effects of different interventions and how they correlate with different biomarkers. Probably the easiest way to do that is as correlatives on much larger studies. But so that's what we're looking forward to doing. And I think, just to answer maybe the second question about cost and scalability. So I think, so I think the per unit cost is several $100s. It's kind of like the razor blade model, where like the microscope is quite expensive and the expertise and all of that. I think the ideal would really be, to do these sorts of medium-sized cohort analyses to really pinpoint what is the most relevant thing. And I would say, like, we profiled 20 different cell types. We have a bunch of computational methods and a lot of sort of fancy stuff. And really the bulk of what I presented could be confirmed at scale with more IHC, quite honestly. So I think just because we have a hammer doesn't mean everything has to be a nail, and this could be simplified and scaled with IHC, depending on what the exact marker is.
I think that's great. I think that makes a lot of sense. I had a question, which is just on, it's an incredibly rich data set that you've generated, having both the H&E and then these multiplex immunofluorescence data sets. Have you started to think about how do you put those together? Could you potentially learn patterns and then use H&E to kind of infer or guess at what immunofluorescence markers might be positive?
Yeah, absolutely. I mean, this is like, it's such a good data set for doing transfer learning like that, because you have the ground truth. They're not even an adjacent section, but the exact same section. So yeah, I think that's the other way in which you could think about scaling this, is that the collection of H&E that we have is massive, that already exists. So if we could feel really confident calling these features from H&E, which I would say is difficult without the ground truth, like you know, a lymphocyte, there's no marker that says whether it's a T cell or a B cell or something else without this. So, but maybe that feature exists, it's just not human interpretable. So 100%, yeah, I think we're really excited about that. We just need to see how it works.
Well, I can't wait to hear more about it. I think we have one more question from Shang, which is a question about the immune landscape in tumors with SMAD4 signaling mutation and metastatic colorectal cancer. Have you looked at SMAD4?
Yeah, so our cohort didn't have a lot of SMAD4. I mean, it's only about 10% of patients. I am pretty excited about some analysis that's come out of Ken Lau's group at Vanderbilt, where they did spatial transcriptomics along with many other things, and also found T cell infiltrated and excluded. And in particular, they found the signature of immune exclusion, and part of that was TGF-beta, which really is dysregulated in SMAD4 mutated tumors. So it sort of does beg the question whether you know, the TGF-beta SMAD4 sort of axis is driving this immune exclusion phenotype. Maybe in a larger cohort than you would see an association between those molecular mutations and T cell exclusion, but we just didn't quite have the power to do that.
Got it. Well, I'm going to switch over to ask a few questions for Seth, but please, you know, to the audience, feel free to keep firing questions. Jeremiah's not going anywhere. So Seth, we have a question. Well, here, I'll start with a question by Sandy Simon. You know, is there a difference in the interactome with native FOXO, or is the interaction the same, but it's just not being recruited to the DNA because you need that PAX3 fusion partner?
Yeah, it's a great question. So I think that a lot of those interactions are the same, and we've actually compared PAX3 FOXO1's interactome to the sort of known interactions for FOXO1, and there's a lot of commonality there. But I think part of it is bringing FOXO1 to the PAX3 specific portions of the genome, and that may change the sort of local protein interactions that are already at that place in the genome. But it might also change the sort of structure of the FOXO1 portion of PAX3 FOXO1 that allows for novel interactions.
Got it. And, you know, and flipping that on its head, because you've identified this, you know, this core phenotype, but you also presented this, there's some variation in difference, at least at the RNA sequencing level. So like, what do you think provides that subtle difference then between the two, between, you know, I think it was the NCOA fusions versus the FOXO1 fusions? Yeah,
so there's quite a bit of difference in the different transcriptomic profiles for each of the fusions. And so I think there's novel interactions that are happening for each of the fusions as well. And so I think that it's just the fact that we're getting these different interactions, and we might be slightly changing the local milieu of proteins within the PAX3 sites that are changing the transcript as well. It may also be that the interactions of the different C-terminal fusions are actually driving the PAX3 fusion protein to different localizations within the nucleus, causing different transcription.
That's a great point. Got it. We have a question from Pawan Raut, which is the interactome indicated that FOXO1 recruitment is important for PAX3 FOXO1 fusion. Oh, sorry, function. I'm also wondering if you use the entire FOXO1 or just the part of FOXO1 that is present in the fusion protein that is, you know, exon 2, so downstream of that.
Yeah, so the exogenous proteins that were introduced here were all based on known fusion sequences. So this is just the portion of the PAX3 FOXO1.
Got it. Got it. Thank you. And then we have an anonymous question. It's long, so I'm going to read it out. It is very impactful how you've characterized this common interactome, given that these 410 proteins may be involved in numerous protein-protein interactions and numerous pathways that may not be tumor specific. For example, you know, they point out that hippo signaling is involved in lots of things. How do you confirm that a given protein-protein interaction will result in a function relative to the fusion with a transcriptional outcome? So I I think they're asking how do you separate out what's cancer specific versus normal physiologic roles?
Yeah, so we tried to do that by doing that depth map comparison to try to find fusion positive tumor specific vulnerabilities that were found within the interactome. And so that was one way of doing it. There's other ways of looking at the data that I think might be helpful to find that as well. And so we've looked at different E3 ubiquitin ligases that might be present in the different fusions to try to figure out if we can target the different fusions using different E3 ubiquitin ligases.
Got it. Well, folks, we still have 10 minutes, so feel free to fire away questions. Otherwise, I'm just going to keep asking questions, which I don't know if, you guys can suffer through. Seth, the other thing, I was wondering if you could just comment on working with such a rare disease. You and I both study rhabdomyosarcoma, but the number of models are pretty limited. The number of cell lines are limited. What limits your ability to keep continuing to grow, your understanding or your group's understanding of of fusion positive rhabdomyosarcoma?
Yeah, so as you mentioned, the models are certainly one thing that restricts us. And so I think they're beginning to create databases or I guess biobanks of different fusion positive rhabdomyosarcoma PDX models. And I'm interested in getting in my hands on some of those. As well as you showed here, or as I showed, I reconstituted all these different fusions. And so there's no real cell models for most of those fusions. There's one for the PAX7 FOXL1 fusions, but the rest of them don't really exist. And so it's all has to be created in this sort of lab setting.
And I wonder, you showed that beautiful example of the fusion with WWTR1. Are there additional, partner molecules that you could even just introduce into a skeletal myoblast line or try to create an alveolar rhabdo, being mindful of the long history of challenges in creating, of doing those experiments? Have you thought about kind of taking some of these and just chucking them into a cell?
Yeah, so I think you're talking about trying to engineer different fusions that might do the same thing based on that 410. Yeah, so I actually tried that. It seems that there's some specificity on where the fusion actually occurs within the proteins in order to actually produce a protein that can be expressed. And so I had a lot of trouble actually expressing these sort of monster fusions that I've generated in the lab.
Fair.
So that was a difficulty in those experiments.
Got it. And I don't want to leave Jeremiah out of the question. So I guess I was hoping that maybe we could talk a little bit more about transfer learning and machine learning. Have you thought about also, there have been now kind of methods taking advantage of single cell sequencing data and overlaying them onto multiplexed immunofluorescence. Have you guys investigated any of that kind of work?
Yeah, so I would say we're working on it. It's exactly the type of question that you might answer with a data set like this. So we have some folks in our group who are trying to do this comparison. I would say, like I said, one of the challenges has been with the SciSif, before we could do this Orion one-shot, the H&E that you got at the very back end of this was really significantly degraded. And I know when they engineered the Orion platform that one of the key considerations was to make this as close to clinically compatible grade H&E as possible. So just to reiterate, I think it's the type of thing that can be done, that should be done, that could increase the scale of what we're finding, and we should do it.
Got it. And going back to Seth, Corinne Lenardic asked a very difficult question. Seth, do you think that the cell of origin is the same for all of the fusions?
It is a very difficult question, and not one I know that I can answer. It seems like it might have to be, just because you're producing the same sort of tumor on the back end, but I don't know.
And that gets to a question, and forgive me, I should know this better, but my understanding was that some of the fusions, the patients with some of the fusions tend to do a little bit better clinically than others. I wonder, does any of your interactome data at least give you an idea of, oh, this will be, we see more cell cycle engagement, or maybe these are going to be more aggressive or interact with, bad actor proteins? I don't know. That's probably well, that's not a great question for me to ask, but.
Yeah, so there's some idea in the field, and I think this is still being investigated, but I think the idea is that PAX7 FOXO1 fusions, the patients do slightly better than PAX3 FOXO1 fusion patients. And we see some signs of that just in the rescue, where some of the fusions rescued cell growth better than others. And some of the transcriptional profiles actually point to different signaling pathways for the different fusions. And so I presented here similarities, but there are definitely differences in the output of these proteins as well. Oh, did we lose an on?
Dr. Hudson, you're on mute.
Yeah, sorry, I think I just cut out for a second there. Great.
Yeah.
Sorry about that. I think that was my connection. Well, sorry, Seth, I missed the rest of your response, but I can bug you about that later. We have an additional question from Pao and Rout. Do the cells show transformation when you have different fusions? And if FOXO1 is not present? So if you also removed FOXO1, I think?
So I guess to address this question, we've done some work in Corinne's lab actually, where they've tried to target specifically PAX3 or FOXO1 using shRNAs to the portions of the transcript that aren't in the fusion. And they show that PAX3 and FOXO1 are both somewhat necessary for these cells to grow outside of the fusion. Is that the question that was asked?
Maybe. I think, Dr. Rout, if you want, feel free to fire away a follow-up, or, we're more than happy to touch base afterwards. Well, I am out of questions. Are there things that you have questions for each other, or other things that we can talk about? Other than that,
yeah, I think it's probably a good time to wrap up here. And
that sounds perfect. I was just going to say, Dr. Hudson, is this a good point to wrap up?
Yes, thanks again, Dr. Zimmerman and Walla, for excellent presentations. Thank you, Dr. Patel, for outstanding moderation. I appreciate that. I'd like everyone to join us March 28th for our next Cancer Moonshot Seminar Series, developing immunotherapies for adult and childhood cancers. Drs. Bossi from Children's Hospital and Dr. Wang from Drexel University. Thanks again. That was outstanding presentations. Appreciate your time.
Thanks, everyone.